Hey guys, and welcome back to Now I Know. Today let's continue our discussion with meiosis. Now last video we did talk about mitosis. We have seen cell cycle, and in mitosis we saw in detail about all the phases and what is centromere, what are spindle fibers, what is kinetochore, etc. So I'm hoping by now we have basic about all those points, and if not, just have a look at that video, because here I might not emphasize on all those points as much, because in meiosis there's a lot of things that are happening. We need to understand a lot that is going on in the prophase 1 especially. So in this video I would like to concentrate on the events that are occurring in the meiosis stages, and that actually understanding mitosis first would help you understand meiosis in better way. Okay, so let's begin. Now meiosis is a type of cell division method, right? And it is a cell division method for the gametes or the sex cells, sperm and the egg. Now everything we are going to talk in terms of humans, right? So for gametes, cell division process is meiosis. Why not mitosis, right? Mitosis is also a cell division, but that is specialized only for somatic cell. So why meiosis is needed for especially for gametes? So the answer is over here. We know what happens during sexual reproduction. There is fusion of gametes, right? That's how the production of offspring takes place. Two gametes, sperm and eggs. Sperm fertilizes eggs, gives us a zygote, and we are diploid organisms, so the zygote is going to be 2n, right? Diploid. Once again, briefly, what do we mean by diploid? That means we have how many chromosomes? 46 chromosomes, or we also say 23 pairs of chromosomes. That means all the chromosomes that we have, they are in pairs, and they are in pairs because one set or one pair comes from father and one pair comes from mother. There are two pairs for every chromosome, and that is why diploid, di means two, right? So that is why we are a diploid organism. Alright, now for us to be diploid, it is important that the gametes are actually haploid, because they are fusing together and we are getting the whole of whatever is there, the sum of whatever is there in sperm and egg in the zygote. So if a zygote has to be diploid, the sperm and egg should be haploid. It has to be having only one set of chromosome. Otherwise what will happen? If it is 2n, we will end up with 4n zygote, right? So we are diploid organism, and since we are diploid organism, it is very important that the gametes that we have are haploid. There is only one set of chromosome in sperm and egg in order to give us diploid zygote. So now our body is all diploid. We are diploid, as I said. All these cells in our body are actually diploid cells. So how do we get this haploid sperm and haploid egg from the diploid cell? That is with the help of meiosis. Now the meaning of word meiosis is reduction. The word itself says what we are talking about. We are talking about a cell division that is going to do reduction in what? In number of chromosome. Because at the time of fertilization, what is happening in zygote? The chromosome numbers double. So once we have this zygote which will form an organism, that organism will produce gametes again. Those gametes should be of 1n. It should be only haploid. So again from 2n it should produce n haploid cells, right? From diploid it should produce haploid cells. So it has to have something that causes reduction in the number of chromosome, and that division is meiosis. Otherwise, what will happen with every round of fertilization, with every generation, the chromosome number will be doubled every time, right? Suppose if okay, let's say for example, sperm and egg, they're haploid, they give rise to a diploid zygote. Now again, they will produce 2n gametes. Okay, so it produces a 2n sperm, let's say for example, which fuses with another more 2n egg. What will be the ploidy number in the next zygote now? 4n, because the number is going to be double. Go on, you are doubling the number of chromosome as you go on if you don't have meiosis. So it is very important that we have meiosis for generation of gametes. That is one reason. One more reason we will see as we go on. Okay, so meiosis is specially for gametes. Now we know about germ cell, we know about somatic cell. I hope we have talked about it. Germ cells are the cells that are found in gonads, right? If it is male, it is in testes, and if it is female, it's in the ovaries. And these germ cells, to begin with, they are diploid cells. They undergo process of meiosis to produce haploid gametes. If you are talking about male, it is sperm. If you are talking about female, it is egg. So I think this much of introduction is good enough for meiosis, what it is, where it happens, and why do we need it. Okay, advantage, as I said, one advantage, because we need to maintain the number of chromosomes after every generation. Also one more advantage as we go on, I will emphasize on that. Now in meiosis, when we talk about, we tend to compare it with mitosis. So as I said before, if you have not seen the mitosis video, I would recommend it. Just have a look, because I'm also going to compare it every now and then with mitosis, because that's how you will see the clear-cut difference. So what happens in mitosis? Briefly, let's just have a look. Mitosis happens in somatic cells, right? For all the somatic cells, mitosis is a cell division process where it is equational division. So whatever is there in the parent cell, the daughter cells would have the same thing, and it is happening in somatic cells, which are 2n or diploid. So the daughter cells also would be diploid. So all the chromosomes I have shown here, 2 pairs of chromosomes, or total 4 chromosomes, all of them would be doubled, right? In S phase, replication of DNA will happen. So all the chromosome will be now in copies. They'll be duplicated. They will align at the metaphase plate, and each sister chromatids will be pulled in the opposite poles and will go in the new daughter cell. As simple as that. That is mitosis. So what is different in meiosis now? I just want to show you briefly the key difference between meiosis and mitosis, because that will give you half of the picture, make half of the thing clear in your head. Meiosis will start with germ cell, right? As I said, germ cell is a diploid cell. So of course a cell is undergoing cell division. So in S phase, all the chromosomes would make its copy. So DNA will be doubled. So all the chromosomes would have copied. Two sister chromatids will be there. In mitosis, all the chromosomes are lining up in this way, right? One after the other. So the key difference in meiosis is, instead of chromosomes, all of the chromosomes lining up in this way during metaphase, the homologous chromosomes, the homologous pairs would line up together. Okay, very important thing. Instead of all the individual chromosomes lining up, in meiosis, it is the homologous chromosomes that line up together during metaphase. So if you look at here in mitosis, it is the sister chromatids that are getting pulled. But in meiosis, these are the homologous chromosomes that are lined up. So what will get pulled? Homologous chromosome. So as a result of this round of division, it is the homologous chromosomes that get separated. Alright, we'll come to it all in detail, why, what, and everything. So remember, in meiosis, the first thing is, in the first division, instead of all the chromosomes individually lining up, homologous pairs will line up together. Yeah, you can see over here. Here it is chromosome number 1, 1, 2, and 2. Okay? Instead of that, what happened? Here chromosome 1 and 1 together, chromosome 2 and 2 together. These are the homologous pairs. So now the spindle fibers are attached to these homologous chromosomes rather than sister chromatids. They are attached to homologous chromosome itself. So when the division happens, anaphase and telophase, this is what we end up with. We have a cell, daughter cells, which would have half the number of chromosome, right? Because one chromosome has gone to one cell and another one has gone to another daughter cell. But look at this, look at the contents. They are still in copies, right? Do not make mistake of saying that these are 2n. These are only n, but the content is double. So these are 2c. I'll come back to this 2n, 2c, 4c and all later. But remember, after the first division, after the first meiosis, what we end up is with the cell which have the chromosomes, haploid cell, but all the chromosomes are still duplicated. All the sister chromatids are still attached. So simple enough to understand what should happen now. We don't want this. We want individual chromosomes, right? So second division will occur where sister chromatids would get pulled. Now this is very similar to mitosis, right? We want the sister chromatids to be pulled. So sister chromatids will be pulled or separated during meiosis 2. So meiosis 2 is very similar to mitosis. So you need not worry much about meiosis 2, because now meiosis 2, if you know mitosis, it's going to be very easy. The only difference would be you start with haploid cell, end up with haploid cell. And here instead of 2, there will be actually 4 haploid cell, because look at this. 1, 2, 3, 4. 4 chromatids are there. So you will get 4 haploid cells at the end. Right? Meiosis 1, remember, you separate the homologous chromosomes, and meiosis 2, you separate the sister chromatids. Still, this is just an introduction, brief overview. I want you to understand the difference. And now let's understand in depth what exactly is happening, in meiosis 1, in meiosis 2, especially in meiosis 1 and prophase 1, very important phase of meiosis. Now you know whether it is meiosis or mitosis, the flow is something like this. We have prophase, metaphase, anaphase and telophase. What does each of the phase mean? We have seen everything in detail in the last video. So similarly, meiosis also will have prophase, metaphase, anaphase and telophase. The only difference is meiosis will have 2 rounds of cell division, right? As you saw, why? Because in first one it is only the homologous pairs that are getting separated. We still need to separate these sister chromatids. So there will be 2 rounds of cell division. So the first round would have prophase 1, metaphase 1, anaphase 1 and telophase 1. And the second round would have prophase 2, metaphase 2, anaphase 2 and telophase 2. Simple enough to understand. We need to see all of them one by one. Trust me, it's very easy and very interesting what happens. The main thing you need to focus on and understand is prophase 1 and metaphase 1. Okay, important of all of it. Like the main thing would lie over here. Rest is easy to understand, and if you know mitosis, rest will be just piece of cake. So let's focus a lot on prophase 1 and metaphase 1. Now 90% of the time the cell would spend is behind prophase 1, and it's very interesting and very important phase of meiosis. Okay, so let's begin with that now. So now what happens, during the interphase is what something like this you can see. All the chromosomes all over the place. It's in the chromatin phase, right? Chromosomes, I shouldn't say, it is in the chromatin phase. All the DNA is yet not wound. So this is how it will look after the replication. And this will now enter the prophase. Now if you remember, prophase is a phase where all this chromatin, all the chromatin would condense to form visible chromosomes. And these are duplicated chromosomes. So ideally you should see 2 chromatids, sister chromatids should be there, right? So from a loose chromatin, this is what we should observe. Because this needs to be pulled in metaphase, so it has to be convenient enough for the cell to separate these sister chromatids, right? So it should be in condensed form. That's the work of prophase. That's what we saw. But what happens in meiosis? Meiosis is a formation of gametes. the gametes that are going to form future offsprings, right? Imagine as it is, the DNA is duplicated, as it is just going to get separated and form gametes, right? There's no change in the genetic content. It is as it is passed on. That's not what we want, right? There has to be genetic variation, susceptibility to certain diseases or something which is related to genes, genetic disorders. There will be no change in that. There will be no improvement in that. There will be no variation in genes. There is no evolution if there is no genetic change. So since it is production of gametes through meiosis, it is very important that there is some change in the parental chromosomes. There is some change in the genetic material, right? You understand my point? What I'm trying to say is, before these duplicated chromosomes get separated, and separation happens in where? Metaphase, we know this. So before it gets separated in meiosis, there should be some change in the genetic material. It is a very, very important. We don't want as it is the same thing to be passed on, right? So in case of prophase 1 of meiosis 1, the important event occurring is recombination. Why? We understood now. Why do we need this? It is very important to have some kind of change in the genetic material. So and it has to happen before the separation. So there is no other phase than prophase 1 where it can happen, right? So the important event of meiosis is recombination, genetic recombination, and that happens in prophase 1. Now you can remember only certain terms over here of prophase 1 and need not remember the whole subdivisions, but it is good enough if you know all the subdivisions. Alright, so now let's see what all is happening, and trust me, it's all logical, okay? There's nothing that forcefully you need to remember. Understand it easily. You will understand you'll remember everything. So what happens in prophase, as I said, the motto of prophase is from chromatin, it should condense and form chromosome. But since it is meiosis, gametes should have some variation. There will be a recombination. So what we need to take care is, we need to take care that chromosomes are condensed, recombination is taking place, and the nuclear envelope is dissolved, because spindle fiber will come and attach, right, before metaphase. So you don't want anything in between chromosome and spindle fiber. So you want the nuclear envelope to be dissolved. These are the basic points. If you remember, we talked about it in mitosis. In meiosis, let's look at it in little more detail, since I said, a lot is happening, because very important phenomena of recombination taking place over here. So the first phase, where they actually just start condensing and you will see the chromatids little bit, is called lactotein. Alright, and lactotein means thin thread. So you can see all this thin threads. Now you can't see at this phase all the sister chromatids. You can't see. You can just see one single chromatids stretched out like this, all like thin threads. That is what is lactotein. The very first phase, where it has just started to condense from all this messy mass. You can see only the stretched out chromatids. Okay, you can't see sister chromatids, but you can see chromatids like this. They are very thin threads, and that is why it is called lactotein. The very first thing for the cell to do, simple enough to understand. So now all these chromatids are kind of ready. What happens in the next phase? We want the homologous chromosomes to be separated, right? So the cell needs to bring all the homologous chromosomes together. So in the next phase, all the homologous pairs would come together. Here we have shown 4 chromosomes or 2 pairs. Blue has come from one parent, and this orange has come from another parent. So homologous pair would be one from mother and one from father, right? So they need to come together. The homologous chromosomes need to come together. So that is the next step, called zygoting. Meaning of zygoting is yoked thread, and yoked means something that comes very close together, very close in relationship. What is coming very close in relationship? The homologous chromosomes. So in zygote, homologous chromosome pair side by side. You see this chromosome 1 from mother and father together, and here also chromosome number 2 and 2 from mother and father together. So zygote, first what happened? They just came up like chromatids, and then they started aligning together. What? Homologous chromosomes. Look at this, look at this one pair. What you can see is, now you can see sister chromatids, right? So you can actually see 1, 2, 3 and 4. In this one complex, you can see 4 chromatids, or I can see 2 pairs of chromosomes, right? I can see 2 homologous chromosomes together. So because I can see 4 chromatids here together for this one, or for any other chromosomes, I can say this assembly as tetrad. Tetra means 4, right? So I can see 4 chromatids together, so I will call this as tetrad. Or I can also say I'm seeing 2 homologous chromosomes together, so this is also called bivalent. Bi means 2. What 2? 2 homologous chromosomes. So this assembly in zygote is called either tetrad or bivalent. So it is bivalent because we can see 2 homologous chromosomes together, or tetrad because we can see 4 chromatids together. Now when they line up like this side by side together, this pairing of homologous chromosome is called synapsis. Okay, so 2 pairs are lying together, we call it synapsis. So what you need to remember, in lactotin all the chromatids were formed, all the chromatids from chromatin, it has come to chromatid phase. But in case of zygote, there are important things you need to remember is homologous chromosomes come side by side, they pair together, and once they pair, we are going to call that pairing as synapsis. Okay, that pairing is called synapsis, but the complex looks like either tetrad or bivalent. Now you know why. And for the synapsis is what? Pairing of homologous chromosome. So for synapsis to occur, there has to be something that keeps these homologous chromosomes together, right? There has to be something that is joining these 2 homologous chromosomes. That is by the help of synaptonemal complex. From synapsis, the word has come. Synaptonemal complex, that is a ladder-like complex, something proteinaceous, connection between 2 homologous chromosomes. You can see in black color I have shown ladder kind of structure. That just starts to begin in zygote, and that's how synapsis is occurring. Now this has to complete, right? So when this completes is what we call packaging. Packaging mean thick threads. So when this synaptonemal complex completely is formed throughout this 2 homologous chromosomes. So now what you can see here, a thick thread over here, they have come close together compared to what they were. So this is called packaging, means thick thread. Now all the homologous chromosomes are completely, like I would say hugging each other. They're completely attached to each other. This is the best time for them to exchange the genetic material. And I told you recombination, we need it, otherwise there'll be no variation. So this is the best time. All the homologous chromosomes with each other, they're tightly bound, they are connected with each other. So this is the best time for them to exchange the genetic material. And that happens by crossing over. You might have seen diagrams like that where the crossing over is happening between 2 chromosomes, homologous chromosome. This is one pair and this is another pair. So here somewhere the exchange is happening. Part of this chromosome would be exchanged with this chromosome, and this over here. Crossing over, in simple words, would occur. So you can see here something like this. You can see the crossing over of these homologous chromosome is occurring at these 2 places. So once it is in packaging, genetic exchange between homologous chromosome would take place. Alright, but that genetic exchange is between non-sister chromatids. That means it is between homologous chromosome. It is not going to happen within the same chromosome, but between the homologous chromosome. If I want to show you, this is one chromosome and these are sister chromatids. But if I have a homologous chromosomes, these become non-sister chromatids, right? So the genetic exchange or crossing over is between two non-sister chromatids. It is not between these two, but it is between these two. Alright, and that will be visible clearly in the next phase. That is the diplotin phase. Diplotin means di, means double. That's what you can see now. The exchange, genetic exchange is occurring. And wherever you can see this exchange, it's like crossing over. That place is called chiasma. Okay, wherever you can see this X formation like this, wherever the crossing over is happening over here, over here, over here, that is called chiasma. The meaning of chiasma itself is cross piece. Okay, so wherever you see this cross piece, wherever you see this X shape thing where the genetic exchange is happening, that particularly is called chiasma. Alright, so that's what we will see in diplotin. And once this diplotin is done, once the chiasma is formed, that means the genetic exchange is occurring. The next phase would be dikinesis. Once the exchange is occurred, we want the chromosomes to move apart. So you will see that the centromeres of these homologous chromosomes are actually moving apart. See, here they are very close, but now they will start to move apart. So it will start moving apart. Now the cell is already exchange has occurred. It needs to separate. So nuclear envelope will be broken down. You will see the centrosomes have moved to the opposite poles, and spindle fiber will start attaching itself to the homologous pairs. Okay, not the chromatids, but the homologous chromosomes itself. It will start attaching itself to homologous chromosomes on each side. If you don't have to remember all these substages, the important thing that you remember is, in case of prophase 1, all the homologous chromosomes, they pair together, and that pairing is called synapsis. Now once they pair together, this complex is called either tetrad or bivalent. Once this tetrad or bivalent has occurred, there will be genetic exchange. This genetic exchange is between non-sister chromatids, and as a result of that, you will see X formation wherever the crossing over is occurring, and that is called chiasma. Alright, so these are the important events occurring in prophase 1. At the end of prophase, our chromosomes are ready, which have now got something new. The genetic variation is there. So now let's take the diagram of meiosis, the whole process, and see what happens. So this is our germ cell, which is diploid, that will undergo S phase DNA replication. So you will have all the chromosomes in copies, right? They will be having sister chromatids attached as centromere. What happens in meiosis? First we need to separate the homologous chromosomes, and before they get separate, they should have some variation in them, right? So the first thing is homologous chromosomes would pair together, as we just saw in the prophase 1. Okay, all these events that you can see over here till over here is prophase 1. So homologous chromosomes would pair together. There will be genetic recombination. Wherever that recombination happens, that X shape structure is called chiasma. This is also called tetrad or bivalent because there are 4 chromatids or 2 chromosomes. Now once the exchange has happened, crossing over has happened, this is what new chromosomes would look like. See, so a part of this chromosome has gone over here and part of this chromosome has come over here. This is what is genetic recombination. Alright, now this will undergo separation, and for separation we have metaphase, where all the chromosomes line on the metaphase plate. But instead of single chromosomes lining up, here we will have homologous chromosomes lining up, right? Because we want homologous chromosomes to be separated. So in the metaphase 1, all the homologous chromosomes will line up together. Spindle fibers would be attached to homologous chromosomes instead of chromatids. So this is anaphase 1, right? And after this, what will be there? Telophase 1. Telophase 1, this will be pulled, extremely towards the opposite pole, and nuclear envelope will start forming back. Not going in detail with that since we have talked a lot about it in mitosis. But at the end, so the end product of meiosis 1 is 2 daughter cells that are haploid. See, the reduction has already occurred. We started with 4 chromosomes, but now we have only 2 chromosomes in daughter cells. They are still in copies, but there are only 2 chromosomes. So this part of meiosis Meiosis 1 is where the reduction of chromosome has occurred. So meiosis 1, to be very specific, is called reductional division, because the reduction has occurred in meiosis 1. Reduction of what? Of chromosome. We have half the number of chromosomes than the parental cell. Now it is easy enough to understand they are still, in copies. The copies are still attached. So what we need to do, we need to separate these sister chromatids from each chromosome, and that is what will happen in meiosis 2. Sister chromatids would separate. That is very similar to mitosis. Now once this meiosis 1, the first round, is over, at the end these chromosomes might unwound a little bit, and it will enter the next round, meiosis 2. So the first phase would be prophase 2. Prophase, the same thing will happen, condensation, same thing as in mitosis, okay? No recombination again, no DNA is duplicated, nothing. But the DNA will be condensed, tightly packed chromosomes or sister chromatids would be ready, nuclear envelope will dissolve, spindle fibers will start attaching itself to the chromatids. Next phase would be metaphase 2. What happens? Chromosomes would be at the equatorial plane, at the metaphase plane, and spindle fibers would attach to all the sister chromatids. Followed by anaphase 2, which I have not shown over here, but anaphase 2, what happens? Sister chromatids will be pulled towards the opposite pole, followed by telophase 2, where they are at the extreme ends, extreme opposite poles. Nuclear envelope will form and will have cytokinesis, followed by formation of 4 daughter cells, because now here this each cell will give rise to 2 haploid cells, because the chromatids are getting separated, right? So at the end we will have 4 haploid daughter cell. Now started with diploid germ cell. We had meiosis 1 and meiosis 2 resulted in 4 haploid cell. Meiosis 1 is the separation of homologous chromosome and meiosis 2 is separation of sister chromatids. So now just let me wind up with some key points that you should remember. In case of meiosis, DNA replication occurs only once, but cell division occurs twice, right? That's why you have meiosis 1 and meiosis 2. In case of meiosis 1, it is the homologous chromosomes that get separated, and meiosis 2 is where the sister chromatids get separated. Now since the homologous chromosomes are getting separated in meiosis 1 is the phase where genetic recombination occurs. Very important part. And meiosis 2 is similar to mitosis, because sister chromatids are getting separated. So in meiosis you start with 2N germ cell which is present in gonads, and you end up with 4 haploid cells. Let me just give you a little trick that I utilize to remember this whole thing. One funny example, sometimes I do remember stuff like that when I had no that great understanding about why it is happening, what it is happening. What I used to imagine is this germ cell has been deployed all over its life. Okay, let's take it that way. It has been deployed from the time it was in zygote 1 cell, it got differentiated into germ cell, into gonad. It has been always deployed. That means these homologous chromosomes have always been together. The paternal and maternal chromosomes, they were like best buddies, best friends. Okay, now suddenly they have to undergo meiosis, and these homologous chromosomes needs to be separated from each other. They have been best friends forever. Now they are going to get separated. Before they separate, they just hug each other so tightly and exchange their genetic material as souvenir to remember each other. Okay, that's what is chiasmata. And then they get separated, having part of each other. Sounds funny, but this is how I used to remember. In exam, this came really handy for me. It really worked well. Homologous chromosomes in meiosis needs to get separated. Best friend forever. Have been together always, going to get away from each other, so they will hug each other for the last time. As a souvenir, they will give part of themselves to the other homologous chromosome, to the other pair. Okay, that's what is genetic exchange, chiasma formation, and all that. So that's all. When it comes to meiosis, the important thing is prophase 1, very important, and metaphase 1, where instead of simply duplicated chromosomes, it is the homologous chromosomes that line at the metaphase plate. If you understand this, rest you know it is going to be sister chromatids, rest you know it is going to be easy. And prophase 1 is very important for meiosis 1. I tried my best to explain you why is recombination needed in meiosis 1. So that's all for now. Maybe this video is really long, but I feel it's very important that we understand all the bases of meiosis. Do not get confused, try and understand, go through this video once or twice, read in books how many ever times you can. I'm sure this will, once you understand, it's going to be very easy to remember. And that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.